Assalamu alaikum student. Today the lecture is about the types of enzyme inhibitors. First of all, we can see the simple mechanisms of enzyme actions. We know the substrate is bind to the enzyme at its active site, and as a result, enzyme substrate complex is formed. After the formation of enzyme substrate complex, the substrate is broken down into its product and enzyme is ready to further reactions. The enzyme inhibitors are basically chemical substance that can attach itself with the enzyme in place of substrate. It means that instead of substrate, the enzyme inhibitor it is going to bind to the active site and it will hinder the activity maybe temporarily or partially. So they are called inhibitors, mean to say it inhibit the activity of enzymes. So in this slide, the active sites of enzymes. This one is the active sites of enzymes and the inhibitor is bind, and because of that this enzyme substrate complex it cannot form, and when the enzyme substrate complex it will not form, it means that there will be not formations of the product. So now talking about the type of enzymes inhibitor, there are basically two types of enzymes inhibitors. The first one is reversible inhibitors and the second one is irreversible inhibitors. Now the reversible inhibitors is further divided into the two further types, the competitive inhibitors and non-competitive inhibitors. Let's talking about these types one by one. Reversible inhibitors. Reversible inhibitors, they are actually temporarily hinder the activity of enzymes and making weak linkage with enzymes. They are temporarily hinder the activity of enzymes and making weak linkage with enzymes. It means that they are sometimes attach themselves with the enzyme at the active sites by forming weak linkage, and after sometimes they will detach themselves from the active sites and now the enzymes can perform its activity just like normally. As the name indicates, their effect can be reverse or neutralize partially or completely. How? Because by increasing the substrate concentrations, it means that if you increase the concentrations of substance or a particular solutions, the enzyme is going to attach itself with the substrate and the reversible inhibitors it will release from the enzymes. Now the two types of reversible inhibitors, they are competitive and non-competitive inhibitors. First one is the competitive inhibitors. Competitive inhibitors they have structurally similarity with the substrate. It means that they are going to compete with the substrate because of their structure similarity. And after the competition, enzymes inhibitor complex it will form, but as we know that they are inhibitors, so they will not produce product. The second one is the non-competitive inhibitors. This one is the non-competitive inhibitors. Non-competitive inhibitors क्या होते हैं? दे डू नॉट कम्पीट विद द सबस्ट्रेट. ये सबस्ट्रेट के साथ कम्पीट नहीं करते. क्यों? बिकॉज देयर स्ट्रक्चर्स आर डिफरेंट फ्राम द substrate. They do not have similar structure similarity, however they bind at a place other the active sites, and as a result they can change the shape of enzymes. And when they change the shape of enzymes, it means that the enzymes cannot bind with the substrate. In this slide, the enzymes, this one is the enzyme and this one is the enzymes, and this is the competitive inhibitors. The shape indicate that both have structure similarity. So instead of substrate, these inhibitors will bind the active site, and because of this binding, no product will form, and the substrate will remain as it. Here, the other sides of the in non-competitive inhibitors, this one is the enzyme and this one is the non-competitive inhibitors. In this slide, the non-competitive inhibitors where here it is the substrate, and non-competitive inhibitors it will bind with the enzyme at the site of the other the active sites, but as a result of this binding the shape of the active sites is changed. शेप क्या होगी, चेंज होगी, एंड एज अ रिजल्ट द चेंजिंग ऑफ द शेप ऑफ सबस्ट्रेट इट कैनॉट बाइंड विद द एंजाइम. The last and the second types of enzymes inhibitor is irreversible inhibitors. Irreversible inhibitors are basically permanently inhibitors the enzymes catalysis. They bind the active site by forming a covalent bond or destroying the globular structures of the enzymes and blocking the active site physically. Their effect cannot be reversed because they are irreversible inhibitors and the substrate chaotic concentrations, increasing the substrate concentrations. So that was all about the enzymes inhibitors and its type. Allah Hafiz.